Students, in the previous session of about nutrition in plants, we discussed about the autotrophic nutrition, the introduction to autotrophic nutrition. And in today's session, we will continue this lesson and we will learn about the photosynthesis in detail. So let us start with this session and learn about the photosynthesis. So what are the conditions necessary for photosynthesis? First of all, let's recall that what is meant by photosynthesis. Yes, as we studied that the process of using the energy in sunlight to convert water and carbon dioxide into carbohydrates like starch, or which is usually in the form of glucose, and oxygen released out is called photosynthesis. Now, let us see that what are the conditions which are necessary for carrying out the process of photosynthesis. Yes, the general conditions which are necessary are the presence of a green pigment called chlorophyll, the presence of sunlight, the availability of carbon dioxide and the availability of water. Try to recall the reaction that takes place in the process of photosynthesis. Carbon dioxide plus water in the presence of sunlight and chlorophyll gives starch plus oxygen. So here you can see that in the same way as the reaction was carried out, the materials which were discussed in the reaction are the conditions which are necessary for the photosynthesis. It's chlorophyll, sunlight, carbon dioxide and water. So all these things should be there to carry out this process. So let us now discuss about each of the conditions in detail. So first we will start with the pigment called as chlorophyll present in the green plants. What is meant by a pigment, children? A naturally occurring substance that gives a particular color to a plant or an animal part is known as pigment. And here we are discussing about one such pigment which is present in plants called as chlorophyll. So it is a green pigment and is usually present in the leaves in the structures called chloroplasts. So this is a leaf and green pigment, that is chlorophyll, is present in this structure that is in the leaf. So this is the picture of a chlorophyll, a green pigment. It looks like this tiny particles, tiny molecules present inside the structure of a leaf in green plants. So you can just see that how fine structure it is. So photosynthesis actually occurs inside these spatial organelles called as chloroplasts. And this organelle is found in all the green plants and it is mainly located in the leaves. Hence, we can say that photosynthesis takes place mainly in the leaves. So, we can conclude that plants appear green due to the presence of a substance called chlorophyll in the leaves. And this is the microscopic view of the chlorophyll, how finely it occurs. And this is the internal structure of the chloroplast. So we can now conclude that the conversion of the light energy to chemical energy is actually done by the chloroplast. Now let us discuss about this chloroplast. What is actually present inside the chloroplast? Inside the chloroplast, there is grana, then a lumen, a stroma. Now you can just see the difference. Where is stoma, where is grana, where is lumen? They are present or they are located at a very different place. And thylakoids. And even we can say that chloroplast is a double-layered membrane. Double-layered means it has two membranes. One is the outer membrane and another one is the inner membrane. Outer membrane means the membrane which is covering the inner membrane and also it is covering these entire structure, that is the grana, stroma, thylakoids and lumen. And inner membrane is just beneath the outer membrane. It also protects the internal structure, means all these structures which are present in the chloroplast. So chloroplast is actually a double-layered membrane organelle. That means outer membrane, inner membrane. So now let us discuss about this grana, stroma and thylakoids. What are they and what is it actually meant by? Thylakoid. There are the stacks of discs present inside each chloroplast, and each such disc is called a thylakoid. Let us see again. This is thylakoid. So there are actually the stacks, we can say the stacks of discs, and each such disc is called a thylakoid, and they contain chlorophyll and they help to trap sunlight. That is why green plants are able to make their own food. Now let us define grana. What is grana? Yes, the stacks of the thylakoids are called grana. So these stacks are actually termed as grana, these stacks. And each such disc is called a thylakoid. So this is the difference between a thylakoid and a grana. Now what is actually stroma? Yes, its location is different. The grana are arranged in a fluid matrix which is called stroma. So I hope you are clear with this internal structure of chloroplast. Let us revise again. There are stacks of discs present inside each chloroplast, and each such disc is called a thylakoid. Then the stacks of thylakoid are called grana, and the grana which are arranged in a fluid type matrix is called stroma. So this is all about the internal structure of chloroplast. And as you know that chlorophyll is a green pigment which is present in the chloroplast, which gives the plants green color. And that is why we can say that the plants are green or they appear green due to the pigment called as chlorophyll. So this is one such condition which is very necessary for carrying out the process of photosynthesis. Now let us move on to other condition which is necessary for photosynthesis. First of all, we will discuss about an experiment with which we will prove that one such condition is very much necessary for photosynthesis. So what is that condition? Yes, it is light. Is light necessary for photosynthesis or not? Now let us understand it with an experiment. We have carried out an experiment in which our aim is to find out that light is necessary for photosynthesis or not. So the materials which are needed in this experiment are the green plants, black paper, scissors and clip. So what we will do? How we will carry out this experiment? First of all, we will cover a part of a leaf using strips of black paper. Can you see here? Now we will de-starch the plant by placing it in a dark corner for about 2 to 3 days. That is, we can say just round about 72 hours. Then we will pick a leaf from a de-starched plant and we will test it for the starch to confirm whether it is fully starch free or not. And then we will place the plant in a strong sunlight for at least 6 hours. And finally, we will pick up the leaf covered by the strip of this black paper and we will remove the strip and we will test the leaf for starch. So let us see what type of observation can occur. First of all, we discussed that we covered a part of leaf using strips of black paper, and then we picked up a leaf from a de-starched plant and we test it for starch to confirm that whether it is fully starch free or not. And then we picked up a leaf covered by a strip of black paper and we removed the strip. And now we are testing the leaf for starch. That means we will put a drop of iodine on the leaf. So after some time, what can we observe? The color of the uncovered part of the leaf changes and the covered part, that is also different. You can see here, this is the uncovered part, this and this, and this was the covered part. That means it was covered by the black paper which was stripped. So what is this? This is the covered part. So you can see the difference between the covered part and the uncovered part. Now what can we conclude by this? The conclusion which we see here is that there was no starch formation in the covered part. So this part does not, and that is why this part did not turn blue-black. So the observation which we can see is that the covered portion of the leaf does not turn blue-black. And what can we conclude? That there was no starch formation in the covered part. So this part did not turn blue-black. Instead, it remained brown. And rest of the part which was uncovered, it turned blue-black because there was no starch formation. And that is why this shows that light is very much necessary for photosynthesis. So you can now imagine, and you can even experiment by your own, right? You can pick up a leaf using the strips of black paper. You can cover that part of a leaf, and then you have to keep it in a dark corner where there is no sunlight, so that what will happen? The starch which is actually present, or the starch which has to be synthesized by the green plants, will not get synthesized. After that, you are keeping it in the sunlight by removing that black paper. After that, what will happen? Yes, that the covered part has no reaction and the uncovered part had reaction, and that is why they turned into blue-black. And that is why we can say that light is necessary for the photosynthesis. So that was all about the light, which is necessary condition for the photosynthesis. So it is the sunlight which is the source of the light energy, and that is why it is able to prepare its own food. Now let us discuss about other condition, which is carbon dioxide. It is obtained from the atmosphere through small openings called stomata in the underside of the leaves. And as we saw the internal structure of a chloroplast, in which what happened? In which the chlorophyll acted as a green pigment. And here carbon dioxide is a gas which acts as a very important or the necessary condition for the plant. But how this gas is obtained by the green plants, or is absorbed by the green plants? It is absorbed by the leaves where there are small openings called as stomata, and in singular form we can call it as stroma. So each such stroma is bounded by two half-moon shaped guard cells. Let us see the structure. This is a leaf which is adapted to perform photosynthesis. Why? Because chlorophyll is present, it is also getting sunlight, and it is also absorbing carbon dioxide from the atmosphere. Now what is actually present in the leaf? This is one such leaf in which you can see that we took a part. This is a circle which shows that there is a transportation of these gases taking place. These are the veins of the leaf where this exchange of gases are taking place, like carbon dioxide is taking place. And this is actually present underside of the leaves, the stoma. And each stoma is bounded by two half-moon shaped guard cells, and the guard cells actually regulate the opening and the closing of the stoma. And that is why it is able to exchange the gases. So what can we observe here? That stoma is present underside of the leaves, and it regulates the opening and the closing of the stoma, the guard cells, right? So here are the guard cells which will open and close the stoma. Now let us see what other things happen. In A picture you can see that the stoma are present with the guard cells. Secondly, we can see that the stoma has guard cells, a nucleus, and a stomatal opening. So this shows that the stoma is opened, which is regulated by the guard cells, and it also gets closed by the guard cells. So B is a structure which shows that this is a closed stroma, and C shows the open stroma. So what actually happens when the stroma is closed, the water exits, and when the stroma is opened, water enters. So when there is enough light in water, the guard cells swell up and curve away from each other, and it opens the stroma so that the water can enter. And this allows the carbon dioxide also to enter the leaves, that is the cells of the leaf, right? And when it gets closed, means when there is not enough of light and water, what happens? The guard cells will shrink, and thus they will close the stoma. And now this will allow the water to exit out of the stoma. So these all such things, that is the stoma is regulated by the guard cells. So this was all about the carbon dioxide, which acts as a necessary condition or the factor for carrying out the process of photosynthesis. Now let us discuss about one such experiment in which we can prove that carbon dioxide is necessary. So is carbon dioxide necessary for photosynthesis? Let us understand it with an example or with the help of an experiment. So this is to demonstrate that carbon dioxide is needed or necessary for photosynthesis, and the materials which we need in this experiment is a potted plant, then a solution. Which type of solution? Sodium hydroxide solution. Then a conical flask with a split cork, that means the cork will not be here. Then iodine solution and a dropper. Split cork, here there will be a cork which will close the conical flask. So how we will carry out this experiment? Yes, first of all we will keep the potted plant inside a dark room for few hours when there will be no sunlight, right? So we will keep a potted plant where there will be no sunlight, but we have to keep it watering. Now we will pour the sodium hydroxide solution inside a conical flask, or we can also put the crystals of sodium hydroxide in the flask. And then what we will do? Yes. What will happen when we pour the solution, or if we keep the crystals of the sodium hydroxide solution in the flask? Yes, this will absorb the carbon dioxide of the air. And then we will place one of the leaves without breaking it from the plant inside the flask, and we will cork it. Now when we will cork it, what will happen? There will be no exchange of gases. And now we will keep the entire arrangement out in the sunlight. Now what will happen? After the few hours we will test the leaf inside the conical flask, this flask, and we will also test any other leaf of the plant for starch using the iodine solution. So now let us see that what happens, what can be observed. The observation will be that the leaf that was clasped inside the conical flask does not turn iodine solution into bluish black, and the other leaf turns iodine bluish black. So what is actually happening? What we did, first we potted the plant in a dark room so that the food will not get synthesized because of the sunlight which is not present. Then when we are pouring, or we are keeping the crystals of that sodium hydroxide inside the flask, what will that do? It will absorb carbon dioxide of the air. The plant will absorb carbon dioxide of the air. So it is getting carbon dioxide, it is getting water, but it is not getting sunlight. And you know that sunlight is also very much important. Then we placed one of the leaves inside the flask, and then we corked it. And then we kept the entire arrangement out in the sunlight. And now it is getting the sunlight. So what happened? That the leaf which was clasped inside the conical flask did not turn into iron actually, and that is why it was not able to make the food. It was not able to synthesize the food. And the other leaf turned out to be the bluish black because it caught the sunlight. We also provided it with water and carbon dioxide were also there. We can conclude that the leaf which was present inside the conical flask did not carry out photosynthesis because there was no carbon dioxide available to it. We provided it with sunlight, we provided it with water, but we didn't provide it with carbon dioxide, and that is why there was no exchange of gases taking place when we cocked a part of the leaf. That is, we cocked the leaf in the flask. So when we cocked it in the leaf, that is a part of a plant in the flask, it was not able to exchange the gas. It was not able to get the carbon dioxide. And that is why we can say that carbon dioxide is very much necessary for carrying out the process of photosynthesis. So I hope that you are clear with the experiment. Now let us move on to other important factor which is very much important for carrying out the process of photosynthesis. Water. So this is another important requirement for photosynthesis, in which the root system of plants enables them to obtain water from the soil. So what acts as a very important factor for photosynthesis, for carrying out photosynthesis? It is water. Now how the water will get absorbed by the plant? Yes, there is a root system. The root system consists of a main or a primary root. Then it also consists of the rootlets or the secondary roots and root hair. So these three things are very much important for absorbing the water from the soil. So what happens actually? The root system consists of the primary root, secondary root and root hair, where the root hair increases the surface area of the roots, and they are found in the great numbers near the tips of the roots. So let us see one such picture in which we can show the primary root, secondary root and root hairs. So this is one such picture of a root system. This is the primary root, which is actually present just beneath the shoot system. Then this is the secondary root, that means the root which branches out, and the root hairs. Here are some of the root hairs which are present, which will carry out the absorption of water from the soil. But there are other such tissues which makes it possible for the plants or the roots or the root hairs to absorb the water from the soil. And they are termed as xylem and phloem. So what is actually xylem and phloem? Let us discuss. Water and soluble nutrients enter the root hair, and then they pass through the secondary roots and travel through the main root into the stems and reaches the leaves through structures called as xylem. And once the photosynthesis takes place, starch is formed. That means the food is synthesized. And now this food, that is the starch, is carried out to various parts of the plants through structures called as phloem. So the food is carried out to different parts of the plants through phloem, and water is carried out, water and minerals or the nutrients are carried out to the different parts of the plants through xylem. So always remember this: water and minerals through xylem and food through phloem. So many such different kinds of sugars can be made during photosynthesis, but glucose is the one that is usually made. The glucose is dissolved and transported to the plant via these tissues called as phloem. And this dissolved glucose may be stored for later use, or it is converted into some products such as different sugars, starch, which we are discussing here. It is actually converted into such products called as starch, pectin, etc. So this whole process of photosynthesis is carried out in such way. Water is carried out by this tissue and food is carried out by this tissue. So these both tissues are very much important for the transportation and translocation of water and minerals and food respectively. So this is all about the root system. Let us see one more clear picture of this root hair which carries out this process. This is one such picture of the root hair, and here there is xylem and here there is phloem. So xylem will carry out water and minerals to different parts of the plant, and phloem is the tissue which will carry out food to the different parts of the plant. So this is one such picture of the passage of water which is shown in the roots. So I hope that you are clear with the experiments which proved that some conditions are necessary for carrying out the process of photosynthesis, and I hope you enjoyed this session learning about the photosynthesis in detail. So we are through the autotrophic nutrition. In the next session we will learn about the heterotrophic nutrition. Till then, keep learning, keep enjoying.